So I'm a PhD student in between archaeology and comparative anatomy, and I'm going to be presenting the subject and some preliminary findings for my PhD. So I'm looking at the variation in the morphology of the mandible of micromammals, specifically a wood mouse, Apoddamus sylvaticus, throughout the Holocene in Western Europe, primarily France. And so the Holocene, the objective of my PhD is to see whether or not there have been adaptations to the ecological changes that were imposed by humans. So the original post-glacial ecology of Europe were mature open woodlands with a good biodiversity of plant life and of trees. And then gradually, as humans imposed agriculture on the landscape, it became more arable with open fields, fewer forests, and lower biodiversity. And so the archaeological record shows changes in biodiversity of mammals in response to this ecological change, but there hasn't been very many studies on evolution and whether there's been an evolutionary response. So we're looking at an anthropogenic species which is present before and after the changes imposed by humans, so it didn't just go extinct because of the ecological changes. And the question is whether or not there is a change, an evolutionary change in the morphology, and whether or not that has a function. So why the Holocene? The Holocene is now, it's still happening. So it's all part of the ecology that we're existing in now. And in a period of time, in terms of evolution, 12,000 years isn't very long. So we're looking at that the first graph there is geological time, and then it opens the upper part into another time scale, and then finally a final time scale. And we're looking at the last part of the time scale. And the last small square is represented in this graph, and so we're looking at this point here, which is when the end of the last ice age, so the ice volume goes down. So that's the ecosystem that we exist in now, and we don't have enough data over the last 10,000, 12,000 years to really understand how that has progressed. There's a lot of ecological data over the last 100 years, but systems exist over a longer period of time than that. So I'm looking at increasing the amount of data on ecological niche and microevolution. So, like I said, the original objectives are to see whether or not there's morphological change in response to ecological changes and whether or not that affects function, i.e. the biomechanics of mastication. But first, I need a reference sample of modern specimens in order to create reference data for the biomechanical model and to analyze the morphospace and see whether or not the morphology of the mandible reflects differences in biomechanical function, so that we can infer that function from the morphology of archaeological specimens. So the biomechanical model is just a simple model that simulates bite force based off of data that you can collect from dissection. So I had modern specimens that I was able to dissect to collect muscle data from, and use muscle mass, muscle fiber length, and the insertion and origin coordinates of the muscles to create a calculation that calculates the moment arm of all the muscles added together to simulate bite force. So the origin, it calculates them around the origin, which is the articular, which is that green dot, which is where the mandible attaches to the skull, and then all of the entire structure rotates around that joint. And then you can see here the photograph of the muscle fibers that I dissolved in nitric acid in order to be able to measure them, and a sketch that I did with a microscope in order to measure the insertion and origin areas of the muscles. So you can see the muscle groups that I was dissecting, which connect the mandible to the skull for mastication. So the first data set of modern material that I worked on, I dissected for muscle data, and it's from 2 different sites that are within the same region in France. So they're not too geographically separated, and they each have the 2 species of Epodymus sylvaticus, which overlap geographically, which I could be finding either of these species in the archaeological record. So I wanted to compare them. And the initial data shows that there's does appear to be differences in the muscle maps between the 2 species, particularly in the superior masseter, which is larger in Sylvaticus, and also the temporalis posterior, which is larger in Flavicolis. And the muscle fiber length is consistently longer in Flavicolis, although there isn't any one particular muscle that stands out as being very different. So the model, the bite force is produced by the model. Here I'll explain this. Oh, you test it at different gapes. So as if the mouse was biting down at objects of a different size. And so you're testing to see how much force is exerted on that object. And the different bars are different angles that the food is acting on the tooth, as you can see from the blue arrows. So the 2 species have the same performance at a low gape, and at a large gape, Flavicolis has a more optimized bite. So, yeah. So looking at the fractional participation of the muscles, they seem to reflect what we saw in the muscle mass differences. At the lower gape, where the 2 species have the same performance, the superior masseter is acting much more in Sylvaticus. So we can see that they're using muscles in different ways, even though they have the same performance in terms of bite force. At the larger gape, which has different performance in the 2 species, there is greater action of the temporalis posterior and Flavicolis, which is not surprising since it was a larger muscle. So that is primarily what's causing them to be more optimized at a larger gape. And there are some other interesting things going on as well, like the external pterygoid acts more in Sylvaticus at the larger gape. So it's less optimized at that gape, and it's using different muscles. So it might be compensating there for the fact that it doesn't have the temporalis as developed. And the anterior masseter also acts differently in the 2 species at different gapes. So this wireframe represents the shape change, the greatest shape change within this sample, with both groups pooled together. So the greatest amount of shape change that we're seeing here seems to be around the molar. It's too bad I don't have a pointer. I don't know if I can use this. Yeah. Okay. The molar region is more robust in one direction, and also the coronoid, it's more developed here. In this process, the coronoid process is at a lower angle, and the angular process, part of the ramus, is longer. And this could have clear biomechanical implications because of the muscle insertion areas. So here's the temporalis posterior, and this is where it inserts. So you can see how a larger region there could directly affect the action of that muscle. And there's the rest of the temporalis. And then here is the superior masseter, and this is where the superior masseter inserts. So you can see how the lengthening of this region could affect the insertion of that muscle. And the greater use of the superior masseter could also be reflected in the more robust molar region, because it acts at a lower gape when they might be chewing rather than biting with their incisors. But this shape change doesn't separate the species. So this biomechanical function that we see that describes the greatest shape change is spread across both species. And then when we did a discriminant function analysis, it nearly separated the 2 species entirely. There is one individual that of Sylvaticus that's, Flavicolis that's over with Sylvaticus, but it separates them nearly entirely. So even though the greatest amount of shape variation doesn't separate the species, there is species-specific shape present. And that is represented by the wireframe, which is below the graph. And so what's going on here, to me the most interesting thing is happening to the coronoid. There is once again some changes happening in the angular. But here you see on the flavicolous end of the spectrum, so on this end of the spectrum, the coronoid is more robust and it's more vertical, which would create a better moment arm, which would improve the bite force at a larger gape. And so the second sample that I'm going to talk about doesn't have muscle data, but it is genotyped. So I'm looking at the mandal morphology of these two species over a wider geographic range. And once again, it doesn't separate the overall shape change, shape variation doesn't separate the species at all. And then I've also thrown in one archaeological specimen to show that it lies within the same morphospace, so that we won't be extrapolating when we start trying to interpret or inferring things from the morphology of archaeological specimens. So just looking at PC1, which describes the greatest shape change in this sample, it seems to, I don't have a landmark on the end of the coronoid, but you can see what's happening in that region. But once again, it seems to be doing something like before, where the molar region is becoming more robust, along with the lengthening of the angular process. And over here. And by the way, these wireframes are slightly exaggerated for visualization. So these are not exactly the shapes of the mandibles, but it's a slight exaggeration, but it describes the type of shape change that's happening. And they also don't separate by geography. So the most shape change is not due either to geography or to species. There's the archaeological specimen. And with a discriminant function analysis, it more clearly discriminates the species, and there's no overlap this time. And once again, there seems to be a shift in the angle of the coronoid, with the flavicollis being more vertical. So the preliminary conclusions are that there are biomechanical differences between the two species, which are reflected in the musculature and to a certain extent in the skeletal anatomy, and that these have functional consequences. So one of the species is more optimized at biting at a larger gape. However, the overall shape change are not due to species or to geography. And also the archaeological specimens fall within the morphospace, so that our conclusions based off of modern specimens can be used to infer functional adaptations from the archaeological specimens. And that is an archaeological specimen there. And so the next step is to analyze the archaeological specimens. I've already collected a lot of specimens, primarily using the I2AF database, which has compiled lots of reports from many different sites, so that I can find out who has what species, which is very useful. And so here's an example of some of the specimens that I have from various sites across France, and one from Spain. And so I won't go into all the color coding, but I tried to get sites from different eras and within the various regions that I was collecting specimens from, so that I can get as much chronology from each region as possible. So for example, this is an Iron Age site. This is a Paleolithic site. This is more Iron Age. There's a few medieval sites. That's medieval, and I believe these are Paleolithic. So then looking at the big picture, why is this important? As I mentioned in the introduction, we don't really know very much about the Holocene in terms of ecology, and human-induced landscape changes and biodiversity loss are happening all the time. And it's really useful to know what the effects of human-induced changes are on species and how do they respond in terms of evolution or ecological niche change, which can add another layer of information to our understanding of long-term ecological trends. This is not useful only in a sort of abstract way, as I'm sure everyone here knows, but also that ecological resources are important for our existence. So I'd like to thank various people who I work with and help me. Raphael works on morphology. Sabrina Renaud was the one who lent me all of the modern specimens that I presented here. Cecile Callot worked on the I2AF database. Helder helped me analyze the specimens, and Jean-Claude Marquet donated or lent me lots of archaeological specimens, and these are my 2 units. So are there any questions?
Dans ce que je me souviens, c'est qu'il y avait la taille qui était un facteur extrêmement important pour séparer entre Sylvaticus et Flavicolis, ou même les molaires, les contours des molaires. Donc je veux savoir si vous prévoyez de faire ça aussi et de mettre la taille en relation avec la forme.
Je n'ai pas encore inclus le taille dans l'analyse de discrimination. Aussi, je sais qu'avec les espèces modernes, ça aide. Je dois le regarder, mais aussi je ne sais pas si la taille va changer dans les espèces archéologiques. J'ai essayé de voir si on peut discriminer l'espèce dans les spécimens archéologiques, mais si ce n'est pas possible, j'ai aussi essayé de voir s'il y a la même fonction biomécanique entre les 2 espèces. Donc si on ne peut pas séparer l'espèce pour les spécimens archéologiques, on peut même faire inférence de la fonction de la mandible. Et s'il y a un changement de la fonction de le genre dans le Holocène.
Discriminer des espèces jumelles sur leur forme dentaire, ça fonctionne bien. Ça fonctionne pour des taxons encore plus jumeaux, même entre des populations géographiques au sein d'un taxon type souris. Donc, si tu fais ça sur les dents de tes mulots, je pense que ça va fonctionner. Donc, du coup, tu pourras discriminer tes taxons archéologiques et après, tu pourras faire la biomécanique sur tes mandibules. Tu pourras faire plutôt comme ça.
Avec le deuxième molaire.
La deuxième ou la première. Après, en contexte archéologique, tu auras plutôt de la première. Ouais. Mais du coup, puisque j'ai le micro, je voulais te poser une question. Donc tu cherches des bioindicateurs de l'ouverture du paysage à travers les réponses morphologiques de séries archéologiques. C'est un peu ça l'idée du projet?
C'est plutôt en réponse. Mais aussi, c'est pas nécessaire que c'est à ce moment où il y a une réponse. Et il peut être plus tard. Oui, on ne sait pas en fait à quel temps il va changer ou comment, ou s'il est plutôt un changement de variation, s'il est plus variable dans une époque ou l'autre, et si en réponse de ça.
Cette belle idée, c'est une idée qui est très vaste et pour laquelle la méthodologie, finalement, n'existait pas. C'est ça qui est intéressant, à mon avis, dans ton projet, Lise, si je ne me trompe pas, c'est qu'elle est vraiment en train de construire une approche méthodologique à l'interface de la biomécanique et des spécimens archéologiques, ce qui pose quand même beaucoup de problèmes techniques. Et du coup, le projet s'est évidemment restreint. On va sans doute pas, à la fin de sa thèse, on n'aura sans doute pas une réponse sur les adaptations, mais si au moins sur une espèce, parce qu'au départ il y avait plusieurs espèces, hein, mais là, enfin sur deux espèces, elle arrive à avoir effectivement une méthodologie qui fonctionne. Alors là, on pourra ensuite avancer dans cette direction. Et je dirais que pour moi, c'est une illustration de ce qu'on peut faire dans le cadre du LabEx en mettant ensemble des compétences qui n'avaient pas beaucoup de raisons de se rencontrer, qui aimaient bien se rencontrer, qui discutaient comme ça autour d'une table gentiment, mais qui, sur un même sujet de recherche, pouvaient se compléter. Bon, c'est ça l'intérêt de ce sujet, à mon sens.
On n'a pas utilisé vraiment ces méthodes en archéologie. On a fait des analyses de morphométrie pour discriminer les espèces et pour regarder le développement de domestication, des choses comme ça, mais on n'a pas encore allé au niveau de fonction.
Et en même temps, effectivement, les traits morphologiques qui servent à discriminer ne sont pas nécessairement des traits morphologiques qui ont une signification morphofonctionnelle. Donc, c'est vraiment une approche qui est très nouvelle. And I told that you restricted your research to these two species because it was such a difficult question to find this methodology that you had to restrict.
Oui, aussi, les micro-mammifères peuvent avoir une réaction évolutionnaire très rapide. Donc, on doit récupérer beaucoup de spécimens pour voir le pattern en général, parce que si tu prends 5, ça peut être pas une description de la morphologie de la population. Tu as vu que même entre deux espèces, le morphométrie et le forme morphologique est le même. Donc, il y a beaucoup de variations dans les micro-mammifères. Mais aussi, il n'y a pas aussi. Je pense que c'est intéressant parce que c'est une question écologique. Et il y a beaucoup de questions dans ce qui étudient l'Holocène qui a le niveau de les activités humaines. Et ça, c'est plutôt les humains dans une part d'un système plus grand.
Alors encore 2 questions.
Chez les rongeurs, on ignore, à ce que j'en connais, on ignore fréquemment 2 sources possibles de variabilité, qui sont d'une part le dimorphisme sexuel. Il y a un paradigme, il n'y a pas de dimorphisme sexuel chez les rongeurs, pourtant parfois on peut en voir. Et puis on ignore aussi l'aspect âge biologique du spécimen que l'on regarde. Est-ce que vous avez regardé ces 2 sources de variabilité comme pouvant expliquer au moins en partie la variabilité morphologique?
Les écologistes ou les gens qui collectent les spécimens ne marquent pas si c'est mâle ou femelle avant que c'est préparé. Donc, il n'y a pas suffisant de données pour aller regarder ça. Normalement, dans la description de cette espèce, il n'y a pas une différence entre les mâles et les femelles, même, je crois, en taille. C'est pas marqué là, mais c'est vrai que un des spécimens qui je sais est femelle et a le côté plus robuste. Donc, et le âge de l'espécimen encore, parce qu'ils sont les animaux sauvages, on ne sait pas, mais je peux essayer. J'ai pensé essayer de regarder à l'érosion de les dents, mais encore, il y a beaucoup de choses qui peuvent l'affecter. Peut-être il mange la nourriture plus dure, mais ça aussi peut affecter la morphologie aussi, le sort de terrain ou matériel qu'il mange, qu'il mâche. Donc, malheureusement, on n'a pas ce données, mais ouais.